Welcome dear students. We have started with the transduction topic, and in our last lecture we have discussed the introductory part about transduction. Transduction is the process of genetic material transfer from one bacterial cell to another with the help of the bacteriophage is called as transduction. Bacteriophage, these are the viruses that infect the bacteria. Then we have discussed there are two types of bacteriophage. One is temperate and second is virulent. Virulent phage, they follow the lytic cycle. Okay, they infect the bacterial cell and rupture the bacterial cell, destruct the bacterial cell. That is called as the virulent phage. Temperate phage, they do not disrupt the bacterial cell. Instead, they incorporate the phage DNA is incorporated into the bacterial DNA and then they replicate. Okay. So this introduction we have already discussed in our part first lecture of transduction. In this lecture, we are going to see the detailed process about specialized transduction and generalized transduction. Okay, so these are the types of transduction: specialized and generalized transduction. Specialized transduction is also called as restricted transduction. This is the transduction in which phages transfer only a few restricted genes from one cell to the another cell. That is known as specialized transduction. Okay, so in this specialized transduction, what happens? There is transfer and packaging of small percentage of bacterial DNA with the viral genome. Okay, that is the specialized transduction. मतलब यहाँ पर phage DNA के साथ बैक्टीरियल का DNA भी बैक्टीरियोफाज के कैप्सिड के अंदर पैक हो जाता है. Okay, so that is the special case, hence called as the specialized transduction. What about generalized transduction? This is the transduction in which phages transfer any fragment of the bacterial DNA from one cell to another cell. So in the generalized transduction, there is transfer and packaging of pure bacteria DNA into virus. Okay, so virus like bacteriophage, that is the generalized transduction. जैसे specialized transduction में आप जो phage DNA है, okay, phage DNA is integrated, is incorporated into the bacterial DNA. But in generalized transduction, there is no integration process. Okay, there is no at all integration process. So that is the difference between specialized and generalized transduction. So the efficiency of generalized transduction is low as compared to the specialized transduction. Different bacteria, prokaryotes, the species of the prokaryotes follow the transduction process like D-sulfovibrio, Escherichia, Pseudomonas, Rhodococcus, Rhodobacter, Salmonella, Staphylococcus, Xanthobacter, as well as the Archean like Methanobacterium and Thermoautotropicium. So these are the bacteria which follow the transduction process. Okay, so let's focus on each type, generalized and specialized transduction. We will first discuss about the generalized transduction. In generalized transduction, phage transfer any fragment of the bacterial DNA from one cell to the another cell. Okay, here genetic material is transferred from one bacterial cell to another cell. Example is P1 and P22 phage. These are the examples of the phages. Generalized transduction, it occurs during lytic cycle of virulent as well as temperate phage. So this already we have discussed what is virulent phage and what is temperate phage. So generalized transduction, they follow the lytic cycle. Okay, what is lytic cycle, what is lysogenic cycle, that also we have discussed in our last lecture. Okay, short introduction about the lytic and lysogenic cycle, because this generalized transduction, it follow the lytic cycle, and specialized transduction, it follows the lysogenic cycle, and later on they follow the lytic cycle. Okay, so that is all overall process. After infection, viral genome may or may not integrate in the host chromosome. See, when phage infects the bacterial cell, okay, when phage infects the bacterial cell, the phage genome, it may integrate or not integrate with the host chromosome. Just a specialized transduction में fix है, phage genome is going to integrate with the bacterial genome. Okay, but in the generalized transduction, it may integrate or it may not integrate. Okay, there are both chances. Getting my point? So that is the difference between generalized and specialized transduction. If we will see the actual mechanism of this generalized transduction, then it is similar to that of the lytic cycle. Okay, so let's see diagrammatical explanation of generalized transduction. This is the bacterial cell, which we can call it as a donor cell. Okay, so this circular, it is the bacterial genetic material, bacterial genome, and this is the bacteriophage, and inside this phage, inside the head, capsid head, there is the genetic material. Okay, genetic material is there. So this genetic material inside the capsid has been there. So yes, this phage, it get attached to this, or it get adsorbed to this bacterial cell. Okay, so first step, you can say there is attachment of phage to this bacterial cell. A phage infects a donor bacterial cell. Attachment in cells, we can say that there is, it starts to infect the bacterial cell. Okay, so first step, you can say initiator of infection or attachment of phage to the bacterial cell. In the second step, this phage inserts its genetic material, inserts its genome inside the bacterial cell. Okay, this pink color which has been shown, it is the phage genetic material, and this is the bluish color is the bacterial genetic material. Okay, so after insertion of the phage genome, there is fragmentation. Okay, fragmentation of the genetic material. So phage DNA and proteins are made, and the bacterial chromosome is broken down into pieces. So there is fragmentation of the bacterial genome. In the third process, there is assembly. Okay, in short, we can say that phage starts to replicate itself. Okay, phage kya karega? It starts to replicate itself. How it will replicate? It will use host machinery, it will use host enzymes for the replication purpose, and it will increase its population. Okay, so and sometimes phage, inside this head of the phage, there is incorporation of the bacterial genome. Okay, definitely phage is going to incorporate its genome, incorporates its genetic material in the head, but at the same time there is packaging of bacterial DNA also, because in one cell this phage gene is present and even bacterial gene is present. Okay, so at the time of packaging means the genetic material of the phage is been inserted into or is been packed into its head. Okay, that is the packaging process. So occasionally during phage assembly, pieces of bacterial DNA are packed in a phage capsid. Then the donor cell lies and releases phage particles containing bacterial DNA. As I told, during the packaging process, there is insertion of phage DNA also and even bacterial DNA also. Once the replication process has been completed, there is lysis of this cell. Okay, there is lysis of the cell. Phage will move out of the cell and it will ready to infect another cell. Okay, it will ready to infect another cell. So when it infects the another cell, that is the recipient cell. Okay, so automatically from donor cell the genetic material is been transferred to the recipient cell. Okay, so this phage, it infects the another bacterial cell, that is the recipient cell. A phage carrying the bacterial DNA infects a new host cell, that is the recipient cell. Okay, so inside this phage there is the bacterial DNA. This bacterial DNA will get inserted into this recipient cell and there will be recombination process. Okay, so this is the in faint blue color that has been shown a bacterial DNA, and in this pink color the donor DNA is there. Okay, so that is the recombination process, and finally when the recombinant DNA is been formed or recombinant gene is been formed into that cell, it is referred as recombinant cell. So like that, a recombination can occur producing a recombinant cell, okay, with a genotype different from the donor and the recipient cell. एक हाइब्रिड जीन डेवलप हो चुका है डोनर एंड रिसिपिएंट का. सो दिस इज वेरी सिंपल प्रोसेस ऑफ द जनरलाइज्ड ट्रांसडक्शन. Okay, similar to that of the lytic cycle. Actually this generalized transduction, it follows the lytic cycle. Okay, so that was about the generalized transduction. Yes, this is the another diagram in a simple way. Phage has been attached to the cell, then there is replication, increased population, then it bursts out, it's ready to infect another cell, and then there is a recombination, and finally we get the recombinant cell, and that is known as the transduced bacterium. Means what? Transduction process has happened, wo ho chuki hai, so that is the transduced bacterium. Okay, so that was all about the generalized transduction mechanism. Now this generalized transduction is of two types. One is complete transduction and second is abortive transduction. जो अभी हमने mechanism discuss किया, that was the complete transduction. Means what? Here complete recombination has been happened. Okay, एक full and final process है. The genetic material has been transferred from donor to the recipient, and we have got a transduced cell. Okay, so that is the, we can say that is the complete transduction process. Okay, the transduced DNA fragment or the exogenate DNA fragment, exogenate matlab from the external side, that is from the donor. Okay, so how you will define the complete transduction? The definition is very simple. The exogenate or transduced DNA fragment get integrated within the recipient bacterial chromosome, forming the recombinant chromosome. So that is all about the complete transduction. Okay, that the mechanism which we have discussed, that is the example of complete transduction. What about abortive transduction? Now in abortive transduction, the exogenate means your donor DNA. Okay, so that is your exogenate. The exogenate may not get integrated with the endogenate. Okay, बाहर का जो आपका gene है, it may not integrate with the bacterial gene, that is endogenate, and remains in cytoplasm as free particle. Okay, so that is your abortive transduction. Transduction in which the phage genome has no capacity of integration. As I told, there is no integration with the bacterial genome. Okay, and hence उनका खुद का replication होता है. Means what? Bacterial genome will replicate itself and phage genome will replicate itself, but it will use the host machinery as it is depend on the host for the replication purpose. Okay, so that is about the abortive transduction, the difference between complete and abortive transduction. Next process is your specialized transduction. Now what is a specialized transduction? Here there is transfer and packaging of small percentage of bacterial DNA with the viral genome. Okay, मतलब यहाँ पर bacterial genome के साथ viral genome भी insert हो जाता है. Okay, so that is been there. means let me show you. Suppose this is the phage DNA. ओके, ये है आपका phage DNA और ये है आपका bacterial DNA. Okay, so that is the specialized transduction, means your phage DNA is incorporated with the bacterial DNA और फिर इसकी पैकेजिंग होगी into this phage head. Okay, so that is the overall process, you can say. Yes, so it is also called as restricted transduction. Examples are lambda and psi-80 lysogenic phages. This specialized transduction, it follows the lysogenic cycle. Okay, as generalized transduction, it follows the lytic cycle. Specialized transduction, it follows the lysogenic cycle. Okay, this phages integrates their genome in the host genome at specific site, but sometimes under certain inductions they detaches from the host genome. Means here under certain conditions, when conditions are suitable to the phage, it will come out. Okay, so that is about the specialized transduction. During detachment, they take some gene portion of host genome and leaves some of their own genes into the host genome. Such phage is called as the transducing phage. Transducing phage means what? This is the transducing phage. Okay, so that was the diagram of the transducing phage. When such transducing phage contain host gene, it infects another host and then it transfers it to the next host. Okay, after bursting. So that is about the introduction of specialized transduction. So if we'll talk about the lambda phage, its genetic material is linear double-stranded DNA with a single-stranded projections. 12 nucleotide base pair sequence has been present and it has a linear genome. Okay, the genome is a linear double-stranded enclosed in this head. Okay, in this protein capsid, and this lambda genome, it become circularized using its cohesive ends. Okay, this linear genome, it becomes circular by joining its cohesive ends which are complementary to each other. Circular lambda phage integrates in host chromosome by crossing over. Okay, so that has been there. Let's actually study the process of specialized transduction. Okay, so the process of specialized transduction is very simple as that of the generalized transduction. So it starts from the attachment of the phage to the bacterial cell. Okay, so there is attachment of the phage. That diagram is not shown in this following picture. So first step is attachment of phage to the bacterial cell. After attachment, the phage will introduce its genetic material into the bacterial cell. ठीक है, अटैचमेंट के बाद phage अपना genetic material ये release करेगा into the bacterial cell and it will get attached with this bacterial genome. Okay, so this pink color which has been shown, it is the phage genome, and the blue color which has been shown, it is the bacterial genome. Okay, and this stage is called as the prophage. इस stage को हम क्या बोलते हैं? Prophage. Prophage is the state in which phage genome is integrated with the bacterial genome. Okay, that is known as the prophage. So this step is called as the prophage, or this cell is been called as the prophage. After that, there will be a disintegration that includes some bacterial gene. So it will start to multiply. Okay, it will start to multiply and it will produce the second cell. So like that, from the one cell, or we can say from this one genetic material, from one recombinant genetic material, there will be a number of copies. The assembly will be created. Okay, this process is actually called as replication. Okay, the replication will start, replication of this virus DNA with incorporated host genes. Okay, so this is the replication process, and then as the multiplication happens, the assembly will be created, assembly and release of the transducing phage particles, okay, as that of the lytic cycle, because this follows the lysogenic cycle, but later it undergoes the lytic cycle in specialized transduction. Okay, so once the multiplication copies are increased, the cell will lyse, and this phage is ready to infect the other bacterial cell. Okay, and that is the recipient cell. So from donor, the genetic material is been transferred to this recipient cell, and afterwards its genetic material is been introduced. अब इसका genetic material कैसा है? Hybrid genetic material है. Okay, hybrid genetic material means the phage genome is there and the bacterial genome is there. Okay, simultaneously. वहाँ पर क्या था? Generalized में क्या था? Generalized में phage के अंदर एक तो बैक्टीरियल का पूरा जीनोम होगा, नहीं तो phage का जीनोम होगा पूरा. Okay, let me show you. Generalized में क्या था? In the generalized, either in this head of the phage there will be phage DNA or there will be bacterial DNA. Okay, but in the specialized transduction, what happens? In a specialized transduction, there is phage DNA with the bacterial DNA. Okay, उसी के साथ है attach, separate नहीं है. Okay, so that is the difference between generalized and specialized transduction, which you have to keep in mind. Okay, so here they will recombinant. Okay, so bacterial chromosome containing both virus and donor DNA. It is very simple. And this cell, there is crossover to integrate bacterial gene, bacterial chromosome containing donor DNA. So this is the very important step. In this step there is recombination. Okay, recombination between the donor DNA and between this recipient DNA. ये है donor DNA जिसके अंदर फाज का भी जीनोम है और बैक्टीरिया का भी जीनोम है. ओके, तो ये पूरा जीनोम इसमें इनकॉर्पोरेट हो जाता है, और जो येलो कलर में दिखाया है इट इज द जीनोम ऑफ द रेसिपिएंट सेल. ओके, सो दैट इज अबाउट द स्पेशलाइज्ड ट्रांसडक्शन. ओके, सो mechanism of the generalized and specialized transduction, same is the process. Okay, so I hope it might be clear to you both the processes, specialized and generalized transduction. Thank you.